Here, on the behalf of the Department of Zoology, I welcome Dr. Debushi Saha, and today she's going to talk on about lipophoresis on the web lecture series of the Practical Zoology. And I will call on Debushi to introduce her. Debushi, cheers.
Thank you, Shubhamoida. Well, I'm elated to introduce our today's guest speaker. It's my privilege. Well, quickly coming to her academic background, after she completed her master's in chemistry, she worked as a project fellow in Michigan-based project related to arsenic research and health effects. After that, she joined the PhD program in University of Kalyani in Department of Chemistry. Her work was based on trace element geochemistry in environment samples. After that, she also got selected as NPDL, and she worked. She was my senior in Jadavpur University in School of Environmental Studies as a postdoctorate fellow under SERV DST, and she worked under Dr. Tori Rai Chaudhary. She has numerous publications, notably in fuel, water research, environmental science and technology, etc. And she is presently working as faculty in Netaji Nagar Day College. Besides this, she is also involved in several social work. Without wasting any time, I'll just over to you, Devashri. Please take over the virtual platform.
Thank you, Shambhavitra, for introducing me, and good afternoon, everybody. Thank you, the Honorable Principal and HOD of Department of Geology, and also the distinguished members of this program organized by Sarojini Naidu College for Women. Okay, thank you. And now I'm quickly moving to my topics. I have 3 topics. One is paper chromatography of amino acid. Second one is enzymatic activity of trypsin. And third one is enzymatic activity of lipase. Am I audible to all?
Yes.
Yes, you are.
Yes, we can.
Sound is okay?
A bit
Slower, but it's okay.
but fine. It's audible. Yeah.
Okay.
I think there's some problem in the network.
I'm moving to my slide. Is the slide visible to all?
Yes, we are seeing it.
Okay.
Yes.
Okay. Our first topic is paper chromatography of amino acid. first of all, we have to know what is chromatography. Actually, chromatography is a laboratory technique, just is a laboratory technique to separate the mixture of solution. It has two phases. One is mobile phase and another is stationary phase. What is mobile phase? Mobile phase is a mixture of solution or fluid. Any problem? Am I audible to all?
Yes, you are audible.
Yes. Okay. There is two phases. One is mobile phase, another is the stationary phase. What is mobile phase? Mobile phase is a mixture of solution or a fluid that we have to be separated. And another is stationary phase. What is stationary phase? Stationary phase is a material or substance on which the mobile phase has to be separated, or the mobile phase is selectively adsorbed. Okay. In chromatography, the mixture of solution, that is the different constituent of the solution, runs in the chromatogram with a different speed. That is the cause of separation. And the different partitioning between the mobile phase and the stationary phase is the main cause of separation in chromatography. In other ways, we can say that partitioning coefficient of the different components result in the different retention time in the stationary phase that actually affects the separation. Okay. I'm moving to my next slide, that is type of chromatography. There are several types of chromatography which are well known in our laboratory techniques. First is paper. Second, thin layer chromatography, gel, column, ion exchange, gel filtration, gas liquid, and finally the affinity chromatography. These various type of chromatography generally used in the different laboratory for the separation techniques. Then come to our important point, that is paper chromatography. Paper chromatography is just an analytical method to separate the colored materials or substance, and also the substance having the same polarity. By paper chromatography, again, I am repeating that with the paper chromatography or with the help of paper chromatography, we can separate the colored substance, those which have the same polarity, that is the mixture of amino acid, for example. This is the picture of a general setup of a paper chromatography. In paper chromatography, there is three components. One is mobile phase, another is stationary phase, and third one is the paper. Here, the action or role of the paper is to adjust the host stationary phase or to support the stationary phase like water, any other mixture. Okay. In paper chromatography, the mobile phase in solution that travels up the stationary phase due to the action of capillary of paper. Okay. Next and very important term of paper chromatography is the RF value, or the retention factor. Retention factor or RF value is just a ratio. It is a ratio of distance traveled by the solute to the distance traveled by the solvent. Naturally, the RF value just is a fraction, and generally we get the value up to 2 decimal point. The RF value has two limiting cases. If RF value is 0, in this case, we can say that solute remains in the stationary phase. That is, the solute is totally immobilized. And other ultimate value of RF is 1. That is, the solute has no affinity for the stationary phase and travels with the solvent. For example, if we take an example where solute moves 9.9 centimeter and the solvent moves 12 centimeter. What is the value of RF? That is the ratio 9.9 divided by 12.7. That is 0.78 in a round figure. Here, I'm also to mention that the RF value also depends upon the temperature and the solvent used in the experiment. So several solvent offered several RF value for the same mixture of compound. That is the one of the major disadvantage of paper chromatography. Suppose we performed a paper chromatography experiment. After this experiment, this is the, as I show earlier, that this is the general setup of paper chromatography. After this experiment, suppose we got 3 points in the paper. How can we calculate the RF value? For the first one, that is the red one, that is I'm showing by cursor, red one. What is the RF value? 2 divided by 10. Third one is 6 divided by 10. And last one, blue, is 8 divided by 10. Now come to our mobile phase, that we have to be separate, the amino acid. This is the basic structure of amino acid, containing the amine group and the carboxylic group. There are various types of amino acid, that is arginine, glutamic acid, leucine, valine, etc. The variation or various type of amino acid are obtained due to the variation in the R group, but the amine group and carboxylic group are the same. Next, come to the chemicals and apparatus required for the experiment. For this experiment, this is very important. First one is the normal filter paper or workman number 1, anyone. Solvent, that is the mixture of butanol, acetic acid, and water. Third one is the known amino acid, that is the standard solution prepared by taking 2 milligram in per ml in 10% isopropanol is to 0.1 molar HCl. Simultaneously, unknown amino acid is prepared in the same process. Okay. Fifth, very important, Ninhydrin developer. Sixth, beaker, glass, pipettes, tips or capillary tube may be used, pencil and stapler. Now coming to the very important part of our discussion is the procedure. How to proceed the experiment. Wear a gloves and take a rectangular piece of the filter paper. It will be fine if it is 11 centimeter into 20 centimeter. Then take a pencil and draw two lines approximately from the bottom and the top of the paper. Then draw 3 centimeter, 3 circle in the bottom line and maintain the distance of this circle 3 centimeter. In step 3, next step, pipette out 2 microliter of each standard and also the prepared unknown amino acid solution on the appropriate circle, that is the bottom portion of the filter paper. Okay. Let the sample dry for few minutes and repeat the step 3 for 4 times so that each circle contains 10 microliter of each solution. Next, hang the paper on the beaker containing the mobile phase. And here, 2 important things must be noted, that is, bottom pencil line in the spot must be above the liquid level. Okay. And paper, second one, paper should not touch the side of the beaker. Then cap the chamber and allow the chromatograph for 1.5 hours. One and a half hours later, remove the paper from the beaker and allow the paper to dry in the air. Next step is to develop the spot of the amino acid. In this case, spray the Ninhydrin developer on this paper and we'll get the different spot of the amino acid in the paper. And the most important part is the calculation of RF value. As I discussed in previous slide, how to calculate the RF value. RF value is the ratio, just a ratio, distance of amino acid migrate and the ratio, distance of the solvent migrate. This is the general picture setup of a paper chromatography. And after this experiment, suppose we got a spot of amino acid at 2.1 centimeter from the bottom or the origin line and got the solvent movement, that is from origin to solvent, top line that is 2.1. What is the RF value? 2.1 divided by 2.8 equal to 0.75. This is the overall process of how to perform a paper chromatography experiment of amino acid. Okay. Can I go to the next topic?
Yes.
Okay. Our second topic is the enzymatic activity of trypsin. Slide is visible to all?
Yes.
Yes, it's a
Here, first of all, we have to know what is trypsin. Okay. Trypsin is a serine protease or enzyme that accelerate the hydrolysis reaction of protein. It is generally found in the digestive system of all vertebrates. Trypsin firstly form in our small intestine in proenzyme form, that is inactive form, and trypsinogen, that is produced in our pancreas, that is the activated form. Trypsin breaks the peptide chain at the carboxyl site of the amino acid, for example, lysine, arginine. The whole process is called proteolysis or trypsinization. I'm showing the example of a reaction of trypsin, where trypsin breaks the protein to peptide and amino acid. Ultimate product is peptide and amino acid. Then we have to understand what is the function of enzyme. Actually, enzyme lowers or decrease the activation energy of a particular reaction. That is, in presence of enzyme for a particular reaction, energy requirement is lower down than in absence of enzyme. I'm showing the picture. For a result, the reaction occurs in an easier mode and also in a faster mode. As I already told that trypsin break the protein into peptide and amino acid. Suppose an enzymatic activity of trypsin performed in a test tube. What is the observation? The observation is the appearance of clear solution at the top of the test tube and a white dense precipitation at the bottom of the test tube. These are the chemicals and apparatus required for this reaction. Water bath of 40 degrees centigrade, 0.5% trypsin solution, buffer solution of pH 7, 3% milk powder solution, thermometer, test tube, syringe, and measuring flux. That is all for the seed experiment. Then come to the procedural part of this experiment. This is very important for our discussion. Prepare a water bath of 40 degrees centigrade first. Then take 2 test tubes and mark cross at the middle of both the test tubes. Then label the test tube 1 as case and test tube 2 as control. Take 10 ml 3% powder milk solution in both the test tubes with a syringe or micropipette. Then add 2 ml 0.5% trypsin solution in the test tube labeled case, that is the fast test tube. Then add 2 ml buffer solution of pH 7 into the test tube number 2, that is labeled as control. Put the 2 test tube in the water bath for 40 degrees centigrade or 40 degrees centigrade of for half an hour. Then come to the observation and the conclusion part. In case test tube, as I early mentioned, that after the performing of a reaction with trypsin, in this case, in our case test tube, a white precipitation appears at the bottom of the test tube and simultaneously appearance of clear solution at the test tube at the top of the test tube. And what is for the control test tube? There is no significant change in the mixture solution. Okay. And the conclusion of this experiment is the reaction occurs in presence of trypsin in the case test tube, that is containing the trypsin enzyme, and no reaction occur in absence of trypsin enzyme in the control test tube. Okay. That is the discussion of enzymatic reaction of trypsin. Okay. Now I'm moving to my last topics. That is the enzymatic activity of lipase. Slide is visible to all?
Yes, visible.
First of all, we have to know what is lipase. Lipase is also an enzyme. It's just catalyzed the hydrolysis reaction of fat. It is a subclass of esters also. The main role of lipase in the digestion system. Another important role of lipase is the transport and processing of dietary lipids, that is triglyceride, fats, oils, etc. The action of lipase is very specific. In glycerol, the substrate is the glycerol present in lipid. The action of lipase is very specific. For example, human pancreatic lipase, which is the main enzyme that breaks the dietary fats in the human digestive system, and it converts the triglyceride substrate into the monoglyceride and fatty acid. I'm showing the example how to break the lipase triglyceride into monoglyceride and fatty acid. Next reaction is the another production of another fatty acid and the monoglyceride. As I already told, the enzymatic activity, what is the role of enzyme in a reaction? To lower down the activation energy of a particular reaction. And for this case, reaction occur more easier way and more faster way. Then come to the chemicals and apparatus required for this reaction. First, water bath, test tube, thermometer, measuring cylinder, lipase solution, milk solution, phenolphthalein indicator, sodium carbonate solution of 0.05 molar. The most important part of this discussion is the procedure. How to proceed this reaction. Step 1. Prepare a water bath of 30 degrees centigrade. Then take 2 test tubes and mark as 1 and 2. Then take 1 ml lipase solution in the test tube 1 and put it into the water bath. Then take test tube 2 and add 5 drops of phenolphthalein indicator. Okay. Then add 5 ml milk solution to the same test tube by syringe, and color of the solution turns pink. Next, also add 7 ml solution of sodium carbonate solution into the test tube 2. Then place the test tube 2 into the water bath for 20 minutes. Then pour 1 ml of lipase solution of the test tube 1 into the test tube 2. Again, put the test tube 2 into the water bath. The observation after this experiment, the pink color of the test tube 2 becomes colorless, as I am showing into the slide. Just pink color turns to white. And the cause of the color change or the conclusion of this experiment is actually phenolphthalein shows pink coloration around the pH 10, and it changes to color when the pH drops around 8.3. Okay. In milk, lipase change the color of indicator from pink to colorless due to the breakdown of the fat and increase the concentration of the fatty acid. By adding the sodium carbonate solution, actually sodium carbonate acts to drop the pH of this milk solution and changes the color after the formation of glycerol and fatty acid. That is, lipase break the fat of milk to fatty acid and glycerol at the low pH around 8.3. That is the cause of the change of color of the indicator. Okay. Finally, I have completed all the discussion. Thank you all for hearing the discussion and keeping the deep patience. Thank you.
Thank you, Didi.
Shah, and from the department of behalf of Department of Zoology, I thank you for this nice and detailed presentation, which is very much I can understand for all of us. And I request Chromatography to start the question session. And I can
Thank you, Shubhamoy. Thank you, Devasriti, for a wonderful session. You have kept it very crisp. Thank you for keeping it short and simple. Okay, well, I'll come down to the question is, please let me know if I'm audible. The first question coming along way is like, what are the advantages of chromatography overall over other techniques?
Advantage, it is cost effective and it has two sides. We can separate the mixture of solution quantitatively and also the qualitatively. This is the two advantage.
Okay.
But in paper chromatography, we are able to just detect the or presence of different mixture in the solution, not in quantity.
Okay, that is only qualitative. Okay. Shobhada, you're having the next question with you?
Yes. Well, a question has been asked that how do you find the RF value in which amino acid has the highest of our RF value?
It is not clear to me.
How do we find the RF value and which amino acid has the highest RF value? Is it audible?
Which
Amino acid has the highest RF value.
okay. The highest value obtained for leucine, that is 0.73, and next one is isoleucine, 0.72, and for the and lowest is for histidine, 0.11.
Okay. I'm coming to the next question.
Highest value of amino acid comes due to the lowest retention. Cause
Okay.
of lowest retention.
Okay. There is another question like, what is the term developing in chromatography? What is meant by the term developing?
Paper chromatography, especially for the paper chromatography.
Okay.
Because before developing a point, we can't able to calculate the RF value.
Okay.
For calculation of the RF value and getting the points visible in paper chromatography, we are using the Ninhydrin developer.
Okay. Okay.
So, the next question is,
yeah.
how can we prevent the smear plots in paper.
Your sound is not clear to
How
me.
can how can we prevent smear plots in paper chromatography?
Can't hear you. Can't hear you.
Okay, I'll take up the question. He's telling how can smeared spots can be prevented in paper chromatography.
There is a no question, arise of question of submerged spot in the paper chromatography.
Okay.
If we are able to perform the paper chromatographic experiment more efficiently, there is no question of appearance of submerged spot in the paper chromatography.
Why does it happen?
Submerged spot generally comes due to microbial contaminations or any inefficient handling of experiment.
Okay. That is experimental error. Okay. Um, the next question coming along the way is like, what are the general features of solvents that is employed in chromatography?
Generally, what is the general?
Features of solvents.
Solvents, the mixture of solvents that is um, to avoid only generally water or ethanol, methanol, that is the combination of organic and inorganic solvents are generally used for the stationary phase in the paper chromatography. One thing we have to be noticed here, that is no swelling from the solvent part is avoided.
Okay. Shomada, will you take up the next question?
Yes. The next question is how do we require to why do we require to desulfate silverine to purify it? And it's along with the next part is can we use methanol to water as the mobile phase of paper chromatography to purify it?
Shanghamitra, can you please repeat the question for me?
Okay. He's saying like, why do we require to desulfate green to purify it? This is the first part of the question. The second part being, can we use methanol and water as the mobile phase of chromatography to
Purify it.
No. Coming to the second part of this question, methanol and
Water.
water. No, we can't use this type of solution because it's creates swelling, as I mentioned earlier.
Okay.
And first part is
Why do we require to desulfate green to purify it?
no, there is no requirement to purify this paper because paper in paper chromatography, this is one-time use paper. Okay. After this experiment, we discard this paper. There is no question of purified it. Only to be cautious before handling this experiment, just wear a gloves for the contamination of any oil or microbial contamination into the paper.
All right. So we use this thing. We like if we wear hand gloves, that is sufficient enough.
Sufficient enough for that.
Okay. Shubhamada?
Yes. Well, the next question was why there are two RF values for methionine.
Two?
Am I audible? Two RF values for methionine.
Okay. Generally, for pure compound, there is one RF value for a particular one component. If there is any contamination or any impurities in the compound, in the mobile phase, there is a question of another spot or simultaneous second spot in the paper chromatography. This is the one reason. Another reason is the protonation of the amine group in the amino acid. This is the second reason. Okay, maybe.
Okay. Next question coming along the way is can we find the polarity or non-polarity and relative size of amino acid chain through the experiment? Can we do that, or that is simply an assumption?
We can perform with the mixture of solution or mobile phase, only those have the polar nature. Polar nature.
Okay. Polar nature is
Polar
that is
nature.
what is important.
Yes.
Okay. Okay.
Okay.
Yeah.
The next question is, what is mean by biosurfactant property of lipids?
This is, I think
Something
this
biology
is,
related.
yes, it is totally biology aspect, not from my aspect.
Okay.
Actually, biosurfactant is a part of amphiphilic nature of the living cell. It's totally the part of a biology portion.
Okay. Okay. Is there any practical uses of paper chromatography? Any practical uses?
Yes, it is very effective to identify the mixture of solution or presence of mixture in the solution in a very quick time or very cost effective. In small cost, we can perform this experiment very quickly.
Okay. A question is coming like why hydrophobic paper are used in paper chromatography?
Hydrophobic. This is the capillary nature, due to the capillary nature of the paper, as I mentioned in my slide. Capillary action.
Okay. Nothing like hydrophobic paper. Like what is this actually hydrophobic? I mean, it doesn't have affinity towards the paper. Something like that. Is there anything like hydrophobic, hydrophilic paper? Something like that?
You can make the paper hydrophobic by just to drop the paper in a water or in a polar solvent to make it hydrophobic.
Okay. 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 Yes, sir. Shubhamaya.
Just to accelerate the capillary action of a paper.
Okay. Okay. That is the reason. Okay. Got it.
The next question is, which amino acid it is used if intact peptide is treated with trypsin and chymotrypsin?
Shangamitra, can you please repeat? I can't hear.
Okay. Which amino acid would be released if intact peptide is treated with trypsin and chymotrypsin?
Okay. Trypsin generally breaks the peptide bonds, as I mentioned, in which basic amino acid, for example, lysine, arginine, যেগুলো রয়েছে, okay, contribute to the carboxyl group. And in case of chymotrypsin, keeps those peptide bonds which is aromatic amino acid contribute to carboxylic group. For example, tryptophan, etc.
Okay. The next question is being this: is there is any disadvantage of chromatography? Disadvantage of paper chromatography?
Only just to detect the spot or presence of the polar components in a mixture.
Okay. Only polar
No quantitative application is there.
Okay. Shomada?
I believe I do not have any more new questions.
I'm requesting that I have a next session from 5 p.m.
Yes.
So,
okay. Before you leave, I have got one more question for you. If you can please respond to it, like you already did that. What does it mean if the RF value is 0 and if it is less than 1?
Less than 1. It is obviously less than 1 because it is a ratio. If it is 0, that is immobilized.
Okay, okay.
immobilized.
That
And
is the
if it
reason.
is absolutely 1, that is, it has no affinity for the stationary phase. No affinity.
No affinity for the stationary
Yes,
phase.
completely running with the solvent phase.
Okay, okay. Thank you, Devashri. Shubha Madam, please take over.
Yes, thank you. Thanks, Dr. Shah, for this time you gave for these answers, the questions we had, or the students had. And on the behalf of the department, I again thank you for your presentation and its explanations you have given. And thank you.
Thank you.
Thank you very much, Dr. Shaha.
Thank you, madam.
Our students attended, really enjoyed your presentation. They will benefit a lot from this virtual class. You have answered a lots of questions for the
The
students.
questions, if you have time, the questions will become more questions coming.
Yes, yes,
Sorry,
yes.
Hope to see you
sorry.
again. Hoping
Yes,
to
ma'am.
see you again in the coming
Yes,
Devashri,
future.
absolutely.
uh,
Absolutely.
and I believe you have to come over again because there are a few terminologies which I have also haven't understood that you have to take care actually because this is totally a chemistry part and thank you for that. Really, really,
You
thank
are
you so much
most
for that. Our
welcome.
students will be benefited a lot. Thank you, thank you.
Okay, thank
Come
you.
in our
Okay,
college.
okay, definitely. Thank you. I'm leaving
Thank you,
this
thank you,
lecture
thank
session
you,
with all
Yes.
permission.
thank you,
Okay, thank you
Thank
so
you.
much.
thank you,
Thank you
thank
Please.
so
you.
much.